I wanted to flap my gums a bit more about speciation. We've talked about how it can happen. We've talked about, your allopatric mode, talked about how sympatric speciation can happen and seems to be more common than we used to think. It's worth looking into when and where and how fast it can happen. And one of the really interesting things to study, if you're interested in speciation, is what we call an adaptive radiation. That's a case when you have a founder species that reaches a habitat where there's lots of unoccupied ecological niche space, lots of niches, lots of habitats that no competitors are occupying. When that happens, they can rapidly speciate into a whole bunch of closely related species that occupy slightly different niches. And we call that a species flock. There's one that you're already familiar with because we talked about them in week two, I think, and that's the Galapagos finches. One founder finch would have reached one of the islands roughly 4 million years ago next Thursday and radiated from there into 14 closely related species that nonetheless occupy different niches and, to an extent, different islands. Letting some people in. More classic examples, Hawaiian honey creepers. There's 58, or I should say there used to be 58. Something like half of them are feared to be extinct. But if the Beagle had gotten blown off course and ended up in the Hawaiian Islands, Darwin could have written pretty much the same book, except it would be about Hawaiian honey creepers instead of Galapagos finches, because there are 58 species there that, in the same way as the finches, show adaptations to different diets and different modes of life. There used to be something like 120 species of land snail on the islands of French Polynesia. Unfortunately, on some of those islands, they introduced an African land snail that they were going to raise for food. And the African land snail ate the French Polynesian land snails, which had no defense against it. So many of those are feared to be extinct now. The Hawaiian Islands not only have 58 honey creepers, they've got over 700 species of fruit flies in genus Drosophila and in a closely related genus, Scaptomyza. Restricted to different islands. In some cases, species may be restricted to individual valleys on the same island. And then the biggest one of all is the cichlids. Those are freshwater fish living in lakes in the East African Rift Valley. You may have actually seen some of these because any pet shop with a good freshwater aquarium department is likely to have a tank or two of cichlid species. Many of them are quite beautiful and some of them are suitable for the home aquarium. But there are, or there used to be, about 1,000 of those. Unfortunately, the locals introduced a species into the East African Rift Valley lakes called the Nile perch, which is quite tasty and is an important fishery in the area. Unfortunately, the Nile perch promptly ate the East African Rift Valley cichlids like they were popcorn. So there is, I'm not sure how many of those species are still around. Many of them have been lost. Probably forever. There's actually a good book on them called Darwin's Dream Pond, if you're interested. But let's look at some of these. 2,000 miles from the nearest mainland, you have the Hawaiian Islands, and there's one group of birds in the family Drepananidae that made it over there. They're not huge birds. They don't normally make transoceanic migrations, but somehow their ancestors got to Hawaii and they've undergone an adaptive radiation. Okay, it's 57, not 58 species, more than half extinct or probably extinct. Um, and very much like the finches, they've got different beak types correlated with different food sources, different food supplies. The name honey creeper tells you that a lot of them feed on nectar, but there's certainly plenty that feed or fed on insects or seeds or fruits. And here's 4 of them. The iiwi is one of the most beautiful birds anywhere in the United States. You can see it's got that very long, narrow, curved bill. That's for sipping nectar out of deep, curved flowers. The amakihi, you see, has a narrow bill that's less curved. That one feeds on different flowers. And then a couple of the extinct ones, the o'u and the Kona grobek, you can see they've got much heavier beaks. The Kona grobek was presumably a seed cracker. It's got that big, heavy parrot-like beak. So each of these, in fact, all of 57 honeycreeper species, show adaptations to their environment based on the shape of the beak that they've got, very much the same way as the Galapagos finches do. Come on, Wagner. Okay, East African Rift Valley cichlids. Lake Victoria is one of them. There are 3 big lakes, Lake Victoria, Lake Tanganyika, and Lake Malawi. And Lake Victoria used to contain 300 species of cichlid. That's just freshwater fish in the family Cichlidae, or at least it used to, because the Nile perch was introduced and it ate most of them. And we think we've lost about 200 species of cichlid. There's similar flocks of cichlid species in Lake Tanganyika and Lake Malawi, making 1,000 species in all, or probably less than that now that the Nile perch has killed off many of the species. I'm not sure what the current count is. Again, some of these are popular in the aquarium trade, and you can go see some at PetSmart or Petco on any given day. And they differ in every way you can think of: color, size, and shape, different food sources. There are algae feeders and snail feeders and predators. And do you remember that weird scale-eating fish that showed density-dependent selection? That was an African rift cichlid as well. Sometimes very sophisticated parental care, very complex behaviors. They're an amazing group of fish. So here's where those lakes are. Lake Victoria is right where Uganda, Kenya, and Tanzania meet. And then there's Tanganyika between Tanzania and the Republic of the Congo. And then Lake Malawi between Malawi, Tanzania, and Mozambique. Incidentally, if you're wondering why 2 of those lakes are so very long and narrow, it's because the African continent is very, very slowly splitting along what's called the East African Rift. And those lakes are forming along the rift. That is the zone where someday in the far future, the African continent is going to break open, and East Africa right here is going to end up a big island, which, by the way, is what already happened to Madagascar millions of years ago, 60 odd millions of years ago. But next week, we'll actually talk about how continents rift and how they actually move. So here's a small sample of the 300 cichlid species. You can see they're of different colors. A number of these, by the way, are of males in breeding coloration, and you can actually tell that they are in breeding coloration because they develop these spots on the anal fin, there and there. They're called egg spots. But you can see they've got different proportions, different sizes and directions of mouth, which is correlated with different diets. So if you're picking insects off the top of the water, you'll have a different shaped mouth than if you're scraping algae off the bottom or crushing snails or something like that. The snail crushers have these big crushing teeth in the pharynx, not in the jaws, but in the back of the throat to crush up their snails. Others don't. So there's a small sample of the variety that you get there. And we actually know that Lake Victoria completely dried up 12,400 years ago. And we know that because if you drill into the bottom of Lake Victoria, under the lake sediment and silt, there's a layer of soil with plant remains in it and things like that. And that can be dated to about 12,400 years. That's in the range where you can date this using radiocarbon. So all 300 species of cichlid must have descended from a common ancestor, and they must have done it in less than 12,000 years. In fact, there's a separate little lake that's separate from Lake Victoria called Lake Nabugabo. That lake has only been separate for 4,000 years, and yet it has 5 cichlid species that are unique to itself. So in less than 4,000 years, we've had the appearance of 5 new cichlid species. And this has been proposed as a case of peripatric speciation. I used that word the last time, but remember, peri means around. And the idea is that small isolated populations are likeliest to form on the fringe of a larger one. So if we have the lake level dropping a little bit, what'll happen is the lake will shrink, but we might get some little ponds and small lakes that are isolated around the edge of the large lake. Speciation is likeliest to happen in those ponds. And then if the lake level rises again, the new species and the old will mingle and presumably won't interbreed. On the other hand, we now know, as I've said, that sympatric speciation is surprisingly common. So maybe some of these 300 species of fish arose through processes of sympatric speciation. Lake Tanganyika's had water for over 3.5 million years, and its cichlids are even more diverse in shape and in size. The scale bars for the top ones, that bar is 5 centimeters. The 4 fish below them are smaller because their scale bar is only 1 centimeter. But you can see the body shape and the fin shape and the mouth shape very different for all of these. So we've actually got some numbers on how long it takes new species to form. We have 5 species in 4,000 years in Lake Nabugabo. When we were talking about sympatric speciation, we have the formation of limnetic and benthic species, or very close to species, of stickleback in the past, 12,000 years. We have, 300 Lake Victoria cichlids speciating in 12,400 years. So it looks like we've got a very rough time frame for how long we might expect speciation to take place. The older assumption was that except for very rapid speciation by hybridization and polyploidy, change in species was always going to look gradual. A guy named Richard Goldschmidt brilliant German scientist, heck of a life. He'd been one of the most respected scientists in Germany, with a lab and a whole bunch of assistants, managed to escape the Nazis, made it to the United States, got hired at University of California at Berkeley, and they put him on to teaching freshman biology, which did not go very well because A, he wasn't used to teaching at that level, and B, he couldn't speak English. So if you think some of your professors are a little hard to communicate with, it could be a lot worse. It could have been Richard Goldschmidt. He did recover and rebuild his career in this country before he died in 1958. And he'd suggested that new species and maybe even higher taxa might arise by mutations that have a major effect on phenotype, not just a little small change in shape of a beak or color of the feathers or what have you, but that might actually, seriously rearrange the body plan. This became known derisively as the hopeful monster theory. Most of the time, when you have a mutation that has major effects on the body plan, it ends up in organisms that can't survive. You've got major birth defects going on. And so this was called the hopeful monster theory because when you have macro mutations like this, you get, quote, monsters, but maybe hopefully one of them would actually happen to be really well adapted uh, and survive and become the founder of a new species or even a new higher taxon, new genus, new family, what have you. He probably wasn't right. The very last thing I want to talk about in this class is the relationship between evolution and developmental biology. And Goldschmidt is a great example of what I call Wagner's first law of evolution, which is it's better to be wrong in an interesting way than right in a boring way. And Goldschmidt was wrong, but wrong in a very interesting way. Raised some questions about how exactly genes do build a phenotype. And they were the right questions to ask, and it's only just now that we're beginning to start to answer them. We used to assume as well, not only that change was gradual, but that what was dominant within a lineage was directional trends. That in one generation, I don't know, your birds were going to have a 5 millimeter beak. And in the next generation, they'd have a 5.1 millimeter beak. And then the next generation, 5.2, and then the next generation, 5.3. That change was always very gradual and steady. And we call this anagenesis. The problem is that when you look in the fossil record, you don't see this. What usually you see is that a species will appear, it will seem to persist without a great deal of change for a pretty sizable length of time, and then it seems to disappear. We don't usually see a lot of this directional change. If we don't see this in the fossil record, it's because the fossil record is not perfect. Now, it is completely true that the fossil record is not perfect because we don't have anything remotely resembling all of the fossils that there are. There's lots of once living things that have left no fossil traces worth talking about. I think I was telling some of you folks the fossil record of flatworms you could put into a peanut shell and still have room for the peanut. I, Dr. Ben Wagner, can personally brag of having discovered 1/3 of the world's supply of fossil rotifers. If you know your invertebrate zoology or your aquatic ecology, you've seen rotifers before. They're very common microscopic invertebrates in all sorts of freshwater and even some land habitats. very ecologically important. The total number of fossils that I'm aware of is 3. And I and a gent named Dr. Poinar actually described one important find of fossil rotifers way back in 1994. Um, Not that anybody is really curious about this, but it's a pretty niche field. But hey, I conquered it. 1/3 of all fossil rotifers were discovered by me. Yeehaw! Um, One of my mentors at Berkeley, Jim Valentine, estimated that if you went to a typical shallow water marine habitat, your average beach or mud flat or even coral reef, something like that, only about 13% of the invertebrates would be expected to leave recognizable fossils. So when you find a fossil bed with lots of fossils in it, invertebrates tend to be, in most places, much more common. But even there, you're only seeing less than 1/6 of the total species diversity that used to be there. There's so many completely soft-bodied corals and jellyfish and flatworms and segmented worms and this, that, and the other that you don't have any record of at all. So it is true that we don't have anything like the total number of fossils present, and it can be hard to evaluate speciation using the fossil record. This is what we used to figure that we'd see. On the x-axis, you've got the size of some structure, doesn't matter what it is, and time is graphed on the y. And early in time, you have this structure and it has a mean value right here. And of course, since it's a population, you have some members of the population that are smaller than that mean and some that are bigger and most in the middle. This is a bell curve. And then after some time has passed, you have the population has undergone directional selection and shifted in one direction. And then after a little bit more time, that mean has shifted farther. And here that mean has shifted even farther, and so on. And so evolution seems to be going in a particular direction where the population, the structure size, whatever it is, gets larger and larger with time. The change is in one direction at a pretty steady rate. And we'd call this process anagenesis. And there's a few cases where we do seem to see this. What you're looking at here is on the left, these are different rock layers exposed in East Texas. Actually, some of these you could find in Arkansas if you went looking. So you have a group of rocks called the Wilcox Group, and then the Reclaw Formation, the Weeches Formation, the Cook Mountain Formation, and the Jackson Group right there is the youngest. So time is going from older to younger. And you can see the older fossils of the species called Athleta petrosa. It's a snail. They're smaller and they're less spiny. And they seem to get progressively bigger and progressively spikier over time until we get up here with Athleta petrosa symmetrica, the largest of these fossils with the largest spines. So I guess Athleta petrosa symmetrica is a fossil from the Jackson Group. You want to hear another fossil from the Jackson Group? A, B, C, easy as 1, 2, 3, simple as do, re, mi, A, B, C, 1, 2, 3, you and me, girl. What do you think? Get it? That's a fossil from the Jackson Group. Okay. It's hell telling jokes when I can't actually watch everybody wince. The thrill is gone. A little more detailed study. This is right here on the x-axis. That's the diameter of a portion of the shell of a shell-making protist. It's a single-celled organism, belongs to a group called the foraminifera, specifically a subgroup called fusulinids, very, very common in certain rocks. And the mean diameter of the proloculus changes a little zig and zag, but more or less steadily, going from about 200 microns to nearly 600 microns. And you can see these bars right here. That would be the width of, the width of variation, the width of the bell curve, if you could see it. And you can see that that also shifts as you go from older rocks to younger rocks over a period between roughly 260 and 245 million years. So there are a few cases in the fossil record where we can see what looks like anagenesis. Not so fast, though. A couple of Harvard folks in the 1970s came up with an alternate model for what speciation would look like if you could see it through time. And it's called punctuated equilibrium. Or if you don't like saying the whole thing, call it punk-eq. Punk-eq is the name for the pattern of the timing of speciation that we see in the fossil record. It's just a pattern of timing. It's a hypothesis of rate, how fast speciation happens. You get this confused with, okay, sorry, I've taken a lot of crap from people who have been evidently listening to, too much Hillsong about what punctuated equilibrium is, and they tend to confuse it with the hopeful monster theory, which it is not. They're not the same thing. They tend to claim that it's evidence of scientists completely giving up on Darwinian evolution, which it is emphatically not. And they tend to confuse it with completely different issues, like the ultimate origin of animal life in a time period called the Cambrian, which it is not. Punk-eq just applies to the timing of the origin of species as we see it in the fossil record. And Gould and Eldridge pointed out that when you really do collect a bunch of rocks from different rock layers, sampling a population through time, a species that you collect in an area through time will tend to appear fairly suddenly, persist as it is for some length of time, and then disappear. New species appear abruptly. They stay mostly the same for millions of years. And the predominant evolutionary dynamic is not anagenesis, it's cladogenesis. It's branching. Klados is Greek for branch, as in cladistics. So if we could graph species forming, and let's say on our x-axis we have some measurement of shape, size of an organism or size of a body part or whatever it is, and on the y-axis we have time, gradualistic evolution might look like this. The punctuated equilibrium model would look more like this. What you're seeing here is that species, those long vertical bars represent species that don't change over the length of time that they're around. And the lengths of time that they do change are those branching events, which take very little time at all. They're almost instantaneous. If we put a time axis up here on the y, there's our time axis. Every species in the gradualistic model is changing with time. It's undergoing this anagenetic change. It's doing that. There are gradual trends all over the place. In the punctuated model, all of the change that's happening is happening in these very narrow intervals right there, so narrow that you may not even be able to capture them in the standard fossil record. Now, it'd be great to gather some data on this, and a guy I used to know in grad school actually went and did just that. His name was, and I assume still is, Ross Name. He actually thought about applying for the job that I now have back when we were applying for jobs in 1997, but decided not to apply for it because, quote, I just didn't think I could handle the South. So I got this job and he ended up teaching at, I think, City University of New York, whatever. But while he was in grad school, he studied a group of marine snails called marginellids. This is a typical modern marginellid. They tend not to be very big. They are often, the only real human use for them is in some areas they're made into shell necklaces. So you might find some in souvenir shops and things like that. If they have a common name, they're sometimes called coffee bean snails. And they happen to be really, really common in certain fossil deposits in Costa Rica. So he went down to Costa Rica, lucky stiff, and looked at collections of fossil marginellid shells. And he made a bunch of measurements on these shells. This is his diagram showing the measurements that he made. And there's a technique where you can take a bunch of measurements and combine them arithmetically into a single number that expresses the largest fraction of the total variation in the samples. You can take measurements made on many different axes and grind them down into a single number that expresses the largest possible fraction of the total variance in your sample. You can't really keep track of change in 10 different measurements at once because it's so hard to get 10-dimensional graph paper. Even Amazon was out last time I tried to order some. But you can make, trying to study change in 10 different directions at once is more tractable if you take all those numbers and boil them down into numbers called principal components. So he ran principal components analysis on his marginellids. And here's what he got. This is, again, a time axis with the oldest fossils down here on the bottom and the youngest up here on the top. And this thing, the discriminant axis, that's the same thing as what's called the first principal component. And so he takes all these numbers, and his first principal component is a number that is correlated with shells that are kind of broad in shape. And his population looks like this, and it's pretty close to being a bell curve, maybe a little bit off. But some of the shells are kind of middle of the road, and a few of them are unusually broad, and a few of them are unusually narrow, but most of them are kind of here. You can see here he's got, there's another bell curve. There's another bell curve, and there has not been a great deal of change in the averages until you get over here to this population, and then that one, which actually looks like it might be double humped. And most of the change between the marginellids that are old and the marginellids that are new happens in this time frame, the time frame that he's labeled E, the time frame that corresponds to that zone of rocks. So the transformation is gradual in the sense that we can see a lot of in-betweens, but it's not necessarily steady. We have this period right here for rocks A, B, C, and D where we don't see a great deal of change. And then, of course, in periods G and H, we don't see a great deal of change. And it's only here in the horizons that he calls E and F that we see most of that change happening. So the change is gradual, but it's not at a continuous rate. Most of that change is concentrated in a fairly narrow time span compared with the much longer time spans that these organisms sit around and don't change a great deal. So this is what a transition from one species to the other looks like when you can document it in the fossil record. We can do better than this because we have foraminifera, or forams for short. Forams are either planktonic or benthic. They have bodies that resemble kind of spidery looking amoebas. And there are some naked forams out there, but most of them secrete a covering similar to a shell. Typically, that covering is chalky, made of calcium carbonate. It's calcareous. There's one little group in which it may be silica, and a bunch of species where the covering is made of tiny sand grains or something like that that have been glued together. We say it's agglutinated, and that covering is called a test. Nothing to do with exams, by the way. Testa was Latin for a pot. And somebody who was probably on drugs thought that the shell-like things that forams make looked like little pots. Over there on the left is an actual foram test. We took that picture with the electron microscope years ago. And the test of a foram is perforated with tiny holes. It looks like it just took a shotgun blast. Little extensions of the cell come out of those holes, and the holes are called foramina. Beep, 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 foramina. Beep, beep, beep. Foramen is Latin for a window, so the foramina are the little windows in the test of the foraminiferan, or you can just say foram. The reason you'd want to know about these is forams are extremely abundant in oceans. My major professor is a world expert on forams, and he used to say they were the ideal critters to study because you can find living forams anywhere there's an ocean, and you can find fossil forams almost anywhere you've got sedimentary rocks. And the fact that they're so widespread is what enabled him to travel to something like 160 countries in the course of his career. This is what enabled him one year to be scuba diving on New Guinea tropical reefs, and the next year scuba diving off the waters of Antarctica. There were actually forams living in both places. The man's actually got a Antarctic island named for him. So he used to say that you ought to figure out a part of the world you've always wanted to visit and find what lives there and become a specialist in it. I'm not sure that's great advice for the modern job market, but for anybody thinking about going into research, it is something to consider. One of my other Berkeley professors just so happened to study a group of fossils for which far and away the best examples were found either on the coast of California or the French Riviera. Nice work if you can get it. Anyway, forams that float, sorry, big helicopter outside. Forams that float, they live, they breed, they feed, they die as all things must, and they constantly sink to the bottom. And in some areas, the ocean floor is topped by several feet of what's called foram ooze. It's this mud-like substance where every grain of mud is actually the test of a foram. And foram-rich sediments can build up on the bottom of the ocean many meters thick. And because it builds up very slowly and very gradually in areas that are not very disturbed, we can drill samples of deep sea sediments and bring those up, extract forams, and measure the change from 1 millimeter to the next. So we can look at evolutionary change in extremely fine detail. And when we do that, here's what we get. These sediments are pretty well dated. The scale on the y-axis is millions of years. And between about 10.5 million and 5.5 million, we have a foram species that looks like this. This is the outline of Globorotalia plesiotumida. And you can see right there, that's the size of the test in square millimeters. And you can see that for about 5 million years, there's virtually no change. And then zigzag, we all of a sudden have a great deal of change over the course of roughly 500,000 years. And then we have some zigzagging, but not enormous amounts of change between about 5 million years and the present day. So we've captured the transition from Globorotalia plesiotumida to Globorotalia tumida, which is up here. And so what I want you to see is we do get little bits of zigging and zagging, but the overall pattern is that species tend to stay pretty close to unchanged, maybe a little bit of tracking of environmental change. Certainly, selection is still happening, but most of the change is concentrated in this very narrow time interval. Most of what makes Globorotalia plesiotumida different from Globorotalia tumida happens here. In roughly 500,000 years, which is a very long time from a human point of view, but not very much time compared to the total length of time the Earth has been around, and only a tenth of the length of time that Globorotalia plesiotumida had been around, mostly unchanged. That's punctuated equilibrium. The change is concentrated in these relatively narrow intervals. That's the punctuated part. Outside of those intervals, species tend to stay mostly the same. That's the equilibrium part. So part of the problem is that specialists in different groups of science tend to be talking past each other. If it takes anywhere between, let's say, 10,000 and 100,000 years for a new species to form, if you're working with fossils, that's such a short interval that you often can't see it clearly in the fossil record. If you're working on living organisms, 10 to 100,000 years is unbelievably long. We haven't even had agriculture for 10,000 years yet. 10,000 years ago, your ancestors were hunter-gatherers, basically. Certainly there wasn't anything we'd call civilization. So whether evolution looks gradual or punctuated may depend on your choice of time scale. On the one hand, you can track changes in things like allele frequencies from one generation to the next, year by year, and that looks gradual on that time scale. On the other hand, you can't track year by year changes in most of the fossil record. Trying to track year by year changes using fossils is like trying to time a track meet with a calendar. It's just not the right scale, not the right tool for the job. So that's Gould and Eldridge's point. And I should mention that it's perfectly compatible with the evolution that you've already learned, because we've already seen cases where speciation has happened rapidly, and it tends to be the fastest in populations that are small and isolated. Guess what? The populations that are the smallest and the most isolated happen to be the ones that we are least likely to find as fossils. So it's not surprising that we don't often capture speciation. By the time a population has gotten big, it's large enough that we are likely to find it as fossils, but it's also large enough that it's not that likely to change, because we have, in a large population, you have much less genetic drift. In a large population, you don't really have the founder effect. In a large population, it takes a very long time for a mutant allele to become common. You have slower change in a really big population than you do in small ones. So we don't often capture that transition. Anagenesis is often still thought of as the entirety of evolution. This is a diagram that was created for Time Life books in 1968, and you've all seen various riffs on it. It's one of the most recognizable symbols or icons of evolution, starting with, some knuckle walking apes over there on the left, gradually walking more and more upright and getting shorter and shorter arms, bigger and bigger brains, more and more erect posture, going from Oog the ape man on the left all the way up the march of progress to Fred the naked white guy on the right. Notice that they are all, my major professor actually had a student ask him why ancient humans didn't have penises. And the reason she thought that was that all of these diagrams had been made in such a way that the guy's thighs are hiding his junk. Just so you know, as far as we know, all of these extinct critters did have penises, although there's not much fossil evidence, but probably they did. You probably knew that, but just good to get these things out. Anyway, this is the picture a lot of people have of the way evolution works. One slightly more advanced form replacing the older forms, like the iPhone 5 replacing the iPhone 4, which replaced the iPhone 3, ... moving in a particular direction on an unstoppable trend, in this case from having small brains and knuckle walking to having huge brains and walking fully upright. And of course, this image has been parodied ever since, on everything from album covers from the early 80s to, this rather sad comment on technology, I suppose, that ends with Fred the naked white guy sitting in front of his computer. You can tell this is an old drawing because nobody has CRT monitors these days. But what predominates in evolution is not actually cladogenesis, it's anagenesis. I'm certainly not going to hold you responsible for the detail here, but a more accurate picture of human evolution would look like this, where each of these blue bars is the length of time that a species, as defined by fossils, has been around in the fossil record. And there's lots of them. And they come into existence by a branching process. Evolution makes trees. One of the most important things for people to know, and one of the things that the fewest people seem to know. Now, for what I want to do next in the time that we've got, I'm going to cut over to the whiteboard because I don't have slides for this, but it is important to get this across. So let me see here. Let's go to, there's draw. Yeah, let's do it like this. I get asked every so often, and in fact, at least one of you asked this at the very beginning of the course, when we did that first lab and I was asking for questions and I answered some of them, somebody was asking about why we don't find transitional forms in the fossil record. Well, it's a fair question, but I want to address a misconception about it. Let's say we're talking about the evolution of land vertebrates from fish. People often proceed as if we have this group called fish that we can fit in this box. And in that box, we will put all the fish. And then we have another box that has land life. Let's say it's amphibians or something like that. All right, that's meant to be amphibians. And there was at some time some fish or some species or some lineage of fish that for whatever reason used to be a fish and then quit being a fish, and for a time was maybe both or maybe neither, and then stopped being a fish and started being an amphibian. Like walking from one room to the next. If I were to get up now and walk from my living room into my kitchen, I'd move through my living room. There'd be a time when I'm in the doorway and neither fully in the living room or in the kitchen. And then I would pass through the doorway and I'd be in the kitchen. And people tend to treat evolutionary change like that. Like, here's the fish. Somewhere in between, we have the Darwin fish with the legs. And then once we're through that transition, we have the amphibian. And I guess I just drew a very bad frog. Sorry. You want people with great drawing skills take an art course. That tends to get well, okay. There is a sense in which that's true, in that we do have fossils of critters that had a number of fishy features and fossils of critters that had some fishy features but some amphibian-like features like legs. And so we can sort of trace that, but it doesn't happen anagenetically. Here's what really does happen. If I can get the eraser. There we go. We have this set of organisms, and we can go ahead and call them fish, and we can put them into this bucket right here. There's a pretty kettle of fish. Call that fish. And within that bucket of fish, evolution proceeds by a branching process. Okay, so here are species of fish. They are coming to existence when I'm trying to draw a very crude cladogram right here. And they come into existence like this, and here we can put some over here. Okay, there's lots of fish. Some of our fish start developing new features that we haven't seen before, like maybe fins modified for propping themselves up on the bottom. Maybe some of them even begin kind of strolling out on land for short periods of time, although we now think the earliest amphibian-like critters actually live down on the bottom. And they give rise to a bunch of new species. And at some point, it's actually kind of arbitrary exactly where, we can define a new group and call it, we wouldn't even say amphibians now, we call these tetrapods, critters with 4 legs. Yeah, that's short for tetrapods. And some of those tetrapod features evolved much earlier in evolution, and some of them come in later. But our tetrapods are completely nested within the fish. Our land vertebrates didn't stop being fish, and they didn't lose the basic fish characters from way down here when they started becoming tetrapods. So talking about the transition between fish and tetrapods is a little bit like talking about me going from Conway to my kitchen. That's kind of a weird way to talk about it, but there's no point in my kitchen that is not also in Conway. Actually, I live outside the city limits, so let me put it another way. How would I go from Faulkner County to my kitchen? Well, the answer is that that doesn't make any sense because there is no part of Faulkner County. Well, okay, there's no part of my kitchen that is not also in Faulkner County because I don't live on the county line or anything. It's like asking, how would you get from Arkansas to Conway? Well, it's phrasing the question that way sounds really weird because there's no part of Conway that is not also in Arkansas. Now, if it were Texarkana, it might be a little different, but Conway is pretty firmly nested within Arkansas. And so if we wanted to talk about another let's say how our tetrapods give rise to amniotes, we can nest those within the tetrapods. And then some of those amniotes become mammals, so we could nest those within the amniotes and so on. And all of these things, these organisms are evolving new features and new adaptations and new structures, but not getting away from the old ones. We still have a lot of features that the earliest land vertebrates have. Bony skeletons, for example, vertebrae, many skull bones can match up. We still have a lot of synapomorphies shared with the bony fish that we catch. So when you're talking about a transition from groups, what we often find is a transition into a more specialized version of an existing group. Our ancestors didn't stop being fish when they became distinctive enough that we can label them tetrapods. Our ancestors didn't stop being amniotes when they developed, or our ancestors didn't stop being tetrapods when they became amniotes. So the reason why people say birds are dinosaurs, what happened in that case was you have a bunch of dinosaurs and they diversify and they do all kinds of things, and some become brontosaurus and some become tyrannosaurus and ... One specialized group within the dinosaurs develops a set of new adaptations, feathers and hollow bones and beaks and a short tail with a fan of feathers, develops all of these features, but they don't stop being dinosaurs when they specialize into birds. And that's why, as one of my old professors used to tell people back in the 90s, you eat dinosaur for Thanksgiving dinner, you can go to McDonald's and chow down on some dinosaur McNuggets. And in fact, I can hear the dinosaurs singing outside my front porch, even as we speak. Those little dinosaurs singing off in the woods. Because birds didn't cease to be dinosaurs when they evolved what we think of as, the typical bird definition. That's nested within the dinosaur group. So saying dinosaurs evolved into birds is a little misleading because it makes it, it's misleading in the same way that driving from Arkansas to Conway is misleading, right? It's a weird way to put it. And it's the same kind of thing. Birds are a highly specialized group of dinosaurs, which are a highly specialized group of amniotes, which are a highly specialized group of tetrapods, which are a highly specialized group of fish. We're all still fish. for exactly the same reason that we are still eukaryotes and still chordates. We didn't leave one of those groups when we evolved the features of what we think of as a more specialized group. And it's this, by the same reasoning, humanity, there is no missing link between humans and apes for the simple reason that we're still apes. We have a lot of adaptations. We have bigger brains and more upright posture and greater ability to fuck things up than any other ape species. We are highly adapted for particular lifestyles, and we've developed things like, you know, intelligence and culture, but we did not leave the apes when our ancestors evolved all of those. We are still a specialized group within the apes, within the primates, within the mammals, within the amniotes, and so on. So evolving from ape to man is like going from Faulkner County to my kitchen. We are still a part of everything that it means to say ape. And that's the way to think about transitions between different major groups. You don't stop being what you were when you evolve something new. All right, those are thoughts that have been brewing in my head for a long time, and I feel like they address some of the common misconceptions of how evolution works and the kind of evidence that we look for. We're not really looking for, a critter that is all dinosaur, and then after that something that's 90% dinosaur and 10% bird, and then after that something that's 80% and 20% and so on. What we're looking to do is reconstruct that entire tree. And there's lots of ways to go about that, lots of ways to do it, lots of different lines of evidence that we can draw. But what we're looking at is the entirety of the tree, not just a single set of species that are going in a particular direction, because that is at best only a very small subsample of everything that's going on that we'd want to know about. I'm not sure that has necessarily made things clearer for everybody. I've still got to kind of work that around and definitely find better looking slides, but that's the way that evolution works on a macro scale. And we'll look at some ways we can infer that over the last 2 weeks of the course. All right, by the power vested in me by the state of Arkansas, I'm going to stop the recording.